So our first speaker of the morning, I'd like to introduce Dr. Amir. So Elad got his PhD with Danaper at Columbia, where he did a lot of work on the original scTECH data sets. I think one of his biggest claims to fame or infamy from graduate school was he was the first person to apply two-dimensional t-SNE to flow cytometry data sets. So the ubiquitous t-SNE and VISNE maps that are now in many of these scTECH data sets originated with Elad. After graduating, he did a brief stint in industry, and after that we managed to recruit him back to academia, where he spent a little over a year with the Human Monitoring Center as our lead computational scientist, working on a number of algorithms to facilitate data processing of high-dimensional data sets. He then struck out independently to form his own company, Astrolabe Diagnostics, where he's further developing some of these cool tools. And the project he's going to be talking about to you today is a collaboration we're doing with a large data set we generated at HIMC, where we're leveraging his pipeline to be able to generate some interesting insights from it. Hey, come up, Lud.
Reconnecting. Laura? Is Laura here? Because we need to figure out the Wi-Fi. So while we're waiting for the Wi-Fi to resolve, thank you Miriam and thank you Adib for hosting this event. A couple of months, we've been working on this project for several months now, and about a month and a half ago, Adib came up and asked whether I would like to speak in this symposium. So I asked for more details, and in usual Adib fashion, he said, "Oh, it's going to be a small thing. It's an internal event. It's going to be a handful of people." And then last week, he reaches out and said, "Oh yeah, we're sold out. It's going to be over 100 people." I'm like, "Oh, well done." So a lot of thank you for you guys for organizing this. And I'm going to talk about some of the work we've been doing on developing these comprehensive. Oh, thank you. That was easy. Just refresh. This comprehensive staining data set and the work that we have put into a standardized workflow for acquiring this type of data. And I would say that a big part of the motivation here is developing this infrastructure. Mass cytometry and these other immune monitoring technologies are leaving quite a dent on the research world, and there has been growing interest in employing these kinds of technologies in larger experiments. Hundreds or thousands of samples, experiments acquired over a long period of time. And from my perspective, one of the most interesting and exciting possibilities there are using these ideas of immune monitoring in the context of clinical trials. And some of the potential benefits there are biomarker discovery, segregating responders from non-responders, and identifying co-treatment targets. However, despite the wealth of information and techniques that we have in the context of mass cytometry and other technologies, we're still missing a central workflow that you could plug and play into an existing clinical trial. And Adib has been spending an inordinate amount of time thinking about how to resolve this, and we ended up with this workflow, which I'm going to present here. We decided to apply this pipeline into a novel dataset, a comprehensive antibody staining dataset. The idea was to measure the expression of many different cell surface markers across a wide range of cell subsets in the immune system. In many ways, this mirrors the kind of brochures you can get from BioLegend, where you have this big table and you can examine the expression of every marker on every subset. But we wanted to reproduce this de novo on a single cell resolution. Briefly, the experiment design is as follows. We acquired blood samples from 3 healthy donors, and we stained it using a 21 marker mass cytometry panel that was developed by the Human Immune Monitoring Center. And the goal of this panel is to capture a large number of cell subsets in the immune system, try to be broad as possible, while still leaving some of the mass cytometry channels for in-depth work, which is experiment specific. Afterwards, we wanted to apply some sort of treatment to these samples, and we've chosen to keep it simple and compare a fresh sample versus a fixed one. There has been quite a lot of debate around fixation, especially in the scTECH community, and we already know that some markers are more susceptible to that process than others. We wanted to offer a comprehensive resource people could use when designing their experiments, letting them know whether a specific marker is indeed susceptible to fixation or not. We then used CD45 barcoding to combine all of these samples into a single tube to reduce technical variation. And the next step was to introduce the legend screen. The legend screen is a kit you can get from BioLegend. It is a 496 well plate kit, and in each one of these wells you have a different cell surface marker antibody conjugated to PE. And by introducing anti-PE into your panel, you can measure all of these surface markers in single cell resolution. The next step was to take the legend screen and split it into 38 batches. Each one of these batches was barcoded using a 10 antibody, using a 10 sample barcoding scheme. Overall, we ended up with about 2,200 samples. And acquiring all of this stuff took about 400 hours of cycle time. It was over a month of work at the Human Immune Monitoring Center. I was about to say that this is the largest data set to date. Unfortunately, a bigger one was published a month ago. But it is the largest data set until a month ago, and it is currently the second largest data set with about 63 million events. Now, needless to say, crunching through all these numbers could be a bit of a chore for a human operator. Just debarcoding all of this data could take a few days. So we use the Astrolabe platform. This is a cloud-based platform that is completely automatic. So you can just drag and drop your FCS files, and the platform kicks in and does the analysis. In this case, Astrolabe did the debarcoding, the cleaning, the cell subset labeling, and some of the follow-up statistics. I want to highlight some of the technological tools that have been used in the context of this experiment with the goal of reducing technical variability and batch effects. The first one is employing a lyophilized panel. So the panel, the 21 marker panel that we used, was titrated once and then lyophilized. And we've been using these pellets across the experiment, and in fact, the HIMC has been using them for years. I've been working with several hundred site of experiments by now, and I can say, this is not an understatement, that the lyophilized panels usually have much better data quality than the non-lyophilized ones. Just the fact that you remove the need to titrate and to pipette the panel every time is very conducive for data quality. The second part is using T-tiered barcoding. I cannot imagine running a site of experiment without barcoding nowadays. It is the most effective tool for removing debris and doublets. It's an amazing tool for lowering technical variability. And in this case, the fact that we use the two-tiered barcoding scheme let us use 60 samples in each one of our batches, which was very effective, again, addressing these potential batch effects in the data. And finally, using the standardized automated analysis platform had two main advantages. One, from a time perspective, just manually gating and analyzing this data could have taken days, weeks, maybe even months. Astrolabe could analyze the whole thing, debarcoding and labeling, all of it in about a day. So you can take 2,200 samples, dump them on a machine, and the following day you can start looking at the data and getting insights and ideas on what's going on with your experiment. The second part is, again, reducing that kind of variation that comes from a human operator. And even if you have a single person debarcoding and gating all of this data, they're still going to make decisions along the way. They're going to move their gate 1 pixel to the left, 2 pixels to the right, and so on. And that kind of variability could affect your experiment, especially when data is acquired over a long period of time. By using an automated system, you guarantee that the first sample and the last sample are going to be analyzed in exactly the same way. So the data was acquired, and the first thing we were concerned with is data quality. We examined, as the first goal, we looked at the debarcoding that we've done. And what you're seeing to your left here is a heat map that shows the debarcoding result for one of the batches that were in the experiment. The 5 channels to the left are the palladium channels for the legend screen barcoding. The 4 channels to the right are the CD45 channels for the donor and treatment barcoding. And just by eyeballing it, you see that there is a very clear pattern that shows that the debarcoding worked as intended. I zoomed in on the second heat map from your at the center here, again to show just how clean and crisp the debarcoding process is. One last thing we wanted to do is to compare the automatic debarcoding to manual debarcoding. So we had the debarcode one of the batches manually, and we used that as a baseline to compare the automatic debarcoding again. What you're seeing to your right are 4 statistical metrics that measure accuracy between classification and some sort of baseline. All of these metrics are a value between 0 and 1, where 1 is perfect overlap. And for all four of them, we see that the automatic debarcoding and the manual debarcoding are more or less identical. The next issue we wanted to examine is consistency across the different batches and across the different samples. Again, this was acquired over a month. Things can happen along the way. I'm showing you here one example, and this is the frequency of B cells. At the top figure, each one of these dots is one of the samples, so this is about 2,000 dots. They are separated according to the different donors, and you can see that the B cell frequency across the experiment is amazingly consistent. Again, there's always fluctuations, but overall the donors are the same across the entire sample acquisition. At the bottom of the figure, you see the CD19 intensity in the B cells, and this is by batch. This is not by sample. So there's 38 bars for each one of the samples, and once again, you see that the signal of CD19 across the B cells is amazingly consistent across this month-long acquisition. Here's another example using NK cells. Again, the frequency is to the top. Each one of these dots is a separate sample, and you can see we get the same frequency across the entire acquisition. And likewise at the bottom, the intensity of CD56 in NK cells across different batches. Again, very high consistency. One last issue we wanted to address with regards to this concept of robustness is whether we get a consistent immune profile, whether the three individuals that were in the experiment, the three donors, have the same immune profile across acquisition. To do that, we took the frequency of the different cell subsets in each one of the samples, and we used principal component analysis, a very simple, straightforward dimensional reduction technique to plot this into dimensions. So what we're seeing here are the two principal components. Again, each sample is a dot. They're color-coded according to the donor. And you can see that each donor is its own separate clump of cells, maintaining this immune profile across the entire acquisition. Once we were happy with the quality of the data, we moved into a very classic step when doing flow cytometry and mass cytometry, which is identifying the different cell subsets. Classically, that would be done using manual gating. However, in this case, due to the amount of the data, we used the platform again to do that, the Astrolabe platform. In this heat map to the left, you see the 29 cell subsets that Astrolabe identified. There's a couple of NK cells, multiple myeloids, and several T cells and B cell populations. And for each one of them, you see the median of each one of the markers at the bottom. And the trends here are not surprising. They are what we would expect from this kind of broad profiling data set. The different T cells, naives and memory T cells, and so on. We did drop two subsets from further analysis, the ILCs and the double positive T cells, because when we examined them through our quality control metrics, we could not get a consistent labeling across the different samples. So what you're seeing here are the 29 subsets that remained after quality control. And as with the debarcoding, we wanted to make sure that the automated method is robust and fits what we would expect based on manual gating. So once again, we had to manually gate one of the batches, and what you're seeing here are a score called Matthews Correlation Coefficient, or MCC, for each one of the cell subsets. The MCC is a single cell metric, so it examines all of the single cells in the experiment, and it checks whether there is an overlap between the baseline, which is the manual gating, and the automated gating. Again, it's a value between 0 and 1, where 1 is perfect. We put two arbitrary thresholds, one at 0.9, the jade line. Anything above 0.9 we deem as highly accurate. The second threshold is the gray line between 0.8 and 0.9, which we call it good, but there is some noise there. Anything below 0.8 required further investigation on our end. So we looked into these 6 or so different subsets to figure out what was going wrong. Why is there a disagreement between the platform and between the manual gating? And in all these cases, what we found are what we call thresholding issues. And here is what I mean by that. What I'm showing you here are all of the B cells in the experiment after they were gating for CD38 positive. And all we want to do here is separate between the transitional B cells, which are CD38 high and CD24 positive, and the naive B cells, which are CD38 mid and CD24 negative. In red, you see the naive B cells. In blue, you see the transitional B cells. And to the left is the manual gating, to the right is the platform. And we see that the tip at the top right, where we have the definitely transitional B cells, there's an agreement there. But then there's a no man's land where the manual gating deemed these cells as transitional, while the platform deemed them as naive. And it's a bit of an open question on how to classify these cells. And from our perspective, both answers are correct as long as you do them consistently across the entire data set. And we saw that the 6 cell subsets that were below the threshold we feel comfortable with all had these kinds of questions where one researcher would choose one answer and the other would choose another, and we felt that it's safe enough to continue with these subsets. Now, one of the cool things about the scTECH is that you have a lot of dimensions to work with. And I mentioned that the panel that we had is very broad. It included several markers that are not these classic markers like CD3 and CD19 and CD4, which are very well characterized. There's always an open question of how do these vary across the subsets, the kind of questions that are still open, are still being debated in the scientific community. And in order to address them, we added what we call a profiling level to the labeling process. So the cell subsets I've shown you before, they're all based on rules that are pretty much the consensus of immunology. But here we added this second unsupervised level. And this profiling level starts from the cell subsets that we already identified, but then clusters them using unsupervised clustering using an algorithm called FlowSum. And we take these clusters, we present them to the user, but we do a few things a bit differently than classic clustering. One, if the clusters are highly homogeneous, we just don't display them. We just collapse them into one cluster. And basophils are a good example. Basophils are pretty homogeneous cells, so we can just show them as one cluster. The second thing that we do is we try to guess meaningful names for these clusters. We try to figure out which markers separate these clusters the best and show these to the users as a guide, as a starting point to their inquiry into the data. So overall, we ended up with 71 profiling subsets, which leads us to this very magnificent heat map, which is unfortunately not very readable over a big screen. So instead, we're going to zoom in into a few of the cell subsets and see what Astrolabe came up with. Now, these are unsupervised clusters. Again, the markers that are written here are just a guide. They're not a truth that you should stick to. With that said, it was pretty encouraging to see that the values do make sense. For example, seeing a separation to IgD values in the naive B cells, or seeing CD161 as one of the markers which was chosen for CD8 T cells, going back to this ongoing discussion around mate and mate-like cells. So we ended up with these 71 profiles that we measured over all of these different samples. And now we were ready to actually move on to the goal of this data set, which was to measure all of these legend screen markers across the entire experiment. And we have chosen 2 metrics to present in an aggregate fashion. And just to clarify, all of the single cell data is there, and we are planning on making it available as soon as possible. So if you choose to go into distributions, to go into single cell data, you will always be able to do that. As a resource that includes aggregate statistics, we've chosen to show 2 statistics. And what I'm showing you here is the distributions of anti-PE in 3 different wells of the legend screen. The gray well is blank. It doesn't have any cell surface markers in it. And we use that to establish the background for the signal. And that leads us to the first metric. Any cell which is above the 99th quantile on that background is deemed as positive for the marker. So the first metric we offer is % positive cells within a given profile for a given marker. The second metric is the median intensity. And the median intensity, again, takes the median of the distribution and presents it to the reader, to the user. So overall, we have about 300 antibodies. We have 70 profiling subsets. We end up with 23,000 combinations, and we end up with this, which again is unfortunately not very conducive to being displayed over a big screen. But I hope you can appreciate the fact that this really does cover that typical table I mentioned beforehand in the BioLegend brochure, where you can see all of the markers and many different subsets, and see whether it's positive or not, and what's the median signal for each of them. So we think this could be a very powerful resource. We're going to offer this again on bioRxiv as soon as we can. Meanwhile, let's focus on a few parts of this heat map. And I just decided to go by geography, take something at the top, at the middle, at the bottom, show it to the crowd, and see if there's any interesting anecdotes or observations there. So this is the top. This is a bunch of markers which I'm sure many of you know, many of you have worked with. And here are some of the observations which I found here. And this is entirely anecdotal, just to give you an idea of what these types of resources are capable of. One, CD7 is expressed by NK cells and T cells. And this should not be a surprise to anyone, and it was pretty cool to see that we recapitulated that. So the trend that we expect to see is indeed within this resource. Two, CD5 is not. And it's funny because Adib and I had a Slack conversation earlier this week where he said, CD5 doesn't look right. I was expecting to see CD5 on all of the T cells, but it's only on naive T cells and a few others. And from my perspective, this is a very important warning that anyone who is working with these resources should remember. This is a screen. This was a very broad experiment. It's going to have false positives, it's going to have false negatives. It is a great starting point to the experiment that you want to design, but whatever you're going to do, just validate it with your data beforehand. And I think this is a good example where this is a health truth regarding CD5, but you should be cautious when moving forward with these kinds of resources. The next thing which I found interesting is that there are actually quite a lot of markers which are expressed by all of the cells in the immune system, and this is just one example of them. And as you go down the heat map, I think you can find about 20 or 25 such markers. This is not to say that they're immune system specific, just that they're expressed by everyone. And again, this is just a personal surprise which I found interesting. And now we're moving to the second part of the heat map. This is just the middle. One of the interesting things here is that CD193 is only expressed by basophils. I did not know it's a basophil-specific marker, but apparently it is. One other thing that I want to highlight here is that there seem to be a lot of markers which are expressed by myeloids to some degree, and they seem to be myeloid-specific, and it's quite possible that they are differentially expressed across myeloid. Historically, it seems to me like there has been a lot of emphasis on B cells and T cells, and a bit less work on myeloids for various technical reasons. Based on this, and from my perspective, I think that it's entirely possible that there is some variability there that we're still missing. And it's quite possible that these kinds of resources would be a starting point for deciding on the next marker or the next subset to explore within the myeloid population. And finally, at the bottom of the heat map, again, we're seeing quite a lot of markers that are expressed by myeloids to some degree, with some variability there, which is interesting. And two, there's these patterns that are just like weird. I mean, this marker is expressed by basophils and some T cells, but not all of them, and some myeloids, and maybe by NKT cells as well. I don't know. Evidently, there's quite a lot we still don't know about biology. So here's, again, a starting point and a resource that people could use to answer some of these questions. Taking a step back, I was mentioning that we also have a duplicate of this experiment that was fixated. And we wanted to see what the effects of fixation on some of these markers. And we already have a lot of anecdotal evidence that shows that, for example, CD16 seems to be pretty susceptible to fixation inside of experiments. So we took all the data and we started from about 300 markers. We narrowed it down to about 250 that have some sort of intensity in any of the cell subsets. And then for each one of these, and for each one of the cell subsets we identified, and for each one of the donors, we calculated the ratio between the fixed and the fresh signal. So each one of the antibodies has about 100 data points, 30 cell subsets times 3 donors. We took all of these ratios and we put them on a box plot where the x-axis is the different antibodies and the y-axis is the ratio. So anything above the 0 line, there is a new signal in fixed samples. Anything below the 0 line there was a signal in the fresh sample and that signal disappeared. So we divide them into two groups, a loss of signal and a gain of signal from fresh to fixed. Now, we've arbitrarily put a threshold on times 2 fold change, and after applying the threshold, there are 17 antibodies that lose intensity and 65 that gain some sort of signal between fresh and fixed, which personally I found pretty surprising. It made sense to me that the fixation would lead to a loss of signal. I was pretty surprised to see that there are so many markers that once you fixate, there's going to be a signal there that did not exist before. Is this an artifact? Is this a biological insight? I don't know, but this is something to be aware of when designing these kinds of experiments. Zooming in on some of the data more specifically, we're seeing here a heat map that shows the fold change between fresh and fixed in all of the antibodies that lost a signal. And we see that it is pretty sporadic. Basically, these are cell subsets that had some sort of signal and they lost it. Focusing on some of these distributions, so each one of these figures is the distribution of anti-PE for a given antibody, for a given cell subset, comparing fresh and fixed. So for example, we can see that in NK cells, CD11b is positive within the fresh population, but that signal goes away. Likewise, CD22 in basophils, we were actually surprised that basophils express CD22, but we found some papers from the 90s and early 2000s that show that they do. But that signal goes away as you fixate. And finally, CD62L and CD4 T cells, again, there is some sort of signal in the fresh samples, but as soon as you fixate, that signal goes away. Here is the other heat map, where we are showing gain of intensity between fresh and fixed. And many of these seem to be across the board. So it looks like all of the cell subsets gain some sort of signal as you fixate, which make me think that these are indeed artifacts of the fixation protocol. However, there are a few of them that are more cell subset specific, and it remains to be seen whether this is an artifact or whether fixation helps us unravel some sort of signal that would not be available otherwise. Again, this is a screen, take everything with a grain of salt, and there's a slew of validations that could come up from this work. Focusing on a few of the distributions, to your left you see CCR10 in B cells. The red one is fresh and there's no signal there, but some of the B cells gain some sort of signal after fixation, and the other one is CD140B in CD14 monocytes. Again, no signal before fixation, and there is a bit of it afterwards. So before we move into summary and questions, I'm going to steal 30 seconds of your time for an Astrolabe commercial. And I just want to say that I've been doing this for about a decade now. As Adi pointed out, I'm one of the authors on the TSNE paper, and I'm pretty happy to see that tool being used all over the place. With that said, analyzing side data is still incredibly difficult and time-consuming, whether you have a full-time bioinformatician or not helping you. And it's been my personal mission to liberate my cytometry data, so to speak. We've built this platform to make everyone's life easier. And as I mentioned, we've gone through 2,200 samples in about a day. So if you do need help with analyzing your data, you can either text your name and email to me, or just email me. And also, with the agenda, you've got these pages that say, if you want to know more about Astrolabe, write your name and email and give it to me. So I'm right here and I'm happy to talk about helping with these kinds of analyses. So the summary, we have this comprehensive antibody staining dataset. And as I was saying, we're aiming to release this as soon as possible. All of the FCS files, all of the single cell data, plus all of the summary statistics. We really want this to be as available as possible for everyone. I feel that there's a lot of uncertainty around experiment design, especially with scTECH, and I hope that these kinds of resources would help people feel better about how to set up their experiments. More importantly, or as importantly, we designed this well-controlled experiment design process where really every step along the way we're trying to control as much as possible for technical variability, for any kind of noise that could be introduced by the panel, by staining, and so on. We use an automatic standardized analysis workflow, and I think that as these data sets become bigger and as more data shows up, these are going to become more important, both to save time and to make sure that everyone is analyzing their data in a similar fashion. And overall, this whole design, this whole workflow lets you run experiments that are between tens and thousands of samples. So next time that you sit down to design your scTECH experiment, or honestly any experiment, try to keep these ideas in mind. just to make sure that at the end of the day, the data that you get is going to be as pristine and as accurate as possible. So with that, I will thank you again for your time, and I'm happy to answer any questions. Yes.
Just wondering if some of the effects are from intracellular whether there's some.
So the question was whether some of the fixation effect is due to intracellular scaling as compared to it. Right. So we actually hypothesized a few different reasons that could lead to this, and we are not sure what is the answer. I think that's one of the disadvantages of doing these kind of large screens, that they open up a lot of hypotheses and questions, but they don't really answer them. We are doing a follow-up experiment with 10 of these and trying to figure out what the hell is going on there. But down the line, I would say this is more of a red flag to be aware of when designing your experiment. And when you do reach the point where you want to use one of these antibodies, you will need to answer these kinds of questions. It's mostly about don't just... So the side of it is a very sexy technology. People love using it. It's very exciting for a lot of people. And I found that many groups just throw a bunch of antibodies in a tube and run an experiment, and then things go wrong, and they don't understand why. So this should be a first step. Let's look at the data set, at the database, and see, should we be more careful about things? And the second step is, before we do the big experiment, before we take all of our precious bone marrow samples and run them through the machine, let's get some primary blood and check these markers to answer these kinds of questions. Oh, yes?
Maybe following up on that. But when you get in contact with fixed cells versus other antibodies to that,
so we are going to do a follow-up experiment where we're just doing the same protocol, but instead of using the legend screen, we're going to do it in a regular cytoph panel, just to make sure it's not an effect of the legend screen. Beyond that, I think that we just don't have the bandwidth to go more deeper than that, unfortunately. And we hope that once we release this to the wider scientific community and make it as accessible as possible, people will be able to start picking at these follow-up questions more precisely. A lot of it could also be cell subset effects. Maybe some cells are more sensitive than others to specific fixations, to fixation in specific antibodies. We don't know. We hope that once it's out there, people can help answer these kinds of questions. So the question was, have we considered a protocol where we have multiple levels of staining, some before fixation and some after fixation? And it's fine that you ask that. The first version of this legend screen experiment was very similar, but we actually fixated and then introduced the main panel. And that was extremely problematic because we could not match the fresh and fixed samples. For example, because CD16 disappeared and we couldn't find any CD14, CD16 monocytes in the fixed samples. And the protocol that we ended up doing was, let's stain with the main panel, and then after we're done with that, let's fixate. So I could imagine people going through that process where some of the panel is stained before fixation and some afterwards. I think that many of the people who do use fixation, it's often in the context of tissues where we don't really have much of a choice, and we just want them to make informed decisions on how they design their panel. If a marker is not going to work after fixation, don't use it. But again, the purpose of all these things is not to solve problem, it's just to caution people as they design their experiment. So I can imagine people doing that kind of protocol once they know that they should be concerned about some of their markers. Yes?
Your opinion, how well validated are these less classical markers, or are the antibodies classical markers?
That's a great question, and it is very important to remember that this is not an antibody. This is a clone, and different clones have different behaviors. I mentioned CD22 beforehand. We were very surprised to see CD22 on basophils because when you go into all of the catalogs, you don't see that basophils are supposed to express CD22. You have to dig back in the literature into 1990 something to see that they had an experiment the way they took 4 clones of CD22 and only one of them is expressed by basophils. So I think that you're raising a valid point that maybe it's not a marker thing, maybe it's antibody thing. Maybe these are less commonly used antibodies and the clone that Biolegend provides is highly specific to a given cell subset. We don't know. And again, this is one of the disadvantages of doing a large screen. And by the way, this is not just this large screen. Whenever you see one of these fancy cell papers that had this huge data set that tried to capture everything, these are the kind of questions you want to ask. Like, is this a specific effect for this antibody, for this clone, for conjugating it with PE, for the CyTOF? We don't know. Always validate your own experiments before running the precious samples. Yes?
In terms of like how adaptable is this technology? Like if I wanted another marker, is it easy to use it with your guys?
Super easy. Barely an inconvenience. I can tell you that Sinai is running a lot of experiments using this workflow. And we haven't done it systematically, but sporadically we took data from different Sinai experiments and it matches beautifully, which is something that I do not see with other customers of Astrolabe. And this is why I included this slide here talking about a lyophilized panel and about barcoding and about standardized analysis. If you put these things into your experiment, they're not going to solve everything, but they are going to make your life much easier. And I think that the cool thing about the work that Adib and the Human Immune Monitoring Center has done is that this is really plug and play. This works with tissue, it works with primary blood, with whole blood, with bone marrow, it works with mouse, it works with human tissue, anything.
Everyone, I'm going to wrap up there.
Thank you, everyone.